Hello, everyone. First, I would like to thank the session organizers for giving me the chance to introduce UTAP2. UTAP2 is a user-friendly transcriptome and epigenome analysis pipeline developed by the Bioinformatics Core unit that I'm the head of the unit at the Weizmann Institute. In this presentation, I'll discuss why we developed UTAP2 and go through its main features. Many research questions require the use of NGS technology. UTAP2 provides comprehensive workflows to help researchers, even those without bioinformatics skills, to analyze millions of sequences to insightful results. Providing the means for students and researchers to analyze their data also spares us time at the core facilities to advancements and challenges in the field. So our first version of UTAP2 was published in 2019, and since then has run thousands of times at the Weizmann Institute and has received over 100 citations, highlighting its relevance and contribution. Since 2019, we have continued to improve UTAP, so the version now is UTAP2, and this version includes also epigenome pipelines. So UTAP2 is a web-based interactive interface. Here we see the choices of the predetermined pipelines for the user to choose and run on a cluster, including transcriptome and epigenome pipelines. So presented here, you can see a screenshot of the interface for RNA-seq analysis. The user needs to provide basic information such as the project name, the location of the sequence files, the genome, a location of the output, and an email to receive once the run is done. The user can also perform differential gene expression by selecting the DSeq2 button. Once he does this, basically the samples names is the folder names containing the sequences. He needs to define at least two categories and drag the samples into the relevant categories. We also support including the batch information in order to take it into account. So within the transcriptome pipelines, we support full-length RNA protocols and also 3-end transcript protocols, which include UMI, a unique molecular identifier. The user can also start with a gene count expression matrix. Oops, sorry. Within the pipeline, what happens is if you start with the FASTQ files, the read will be trimmed, mapped to the genome. If you produced a count matrix, either at the level of the reads or at the level of the UMIs, and if she entered the information on differential gene expression, differential gene expression will be executed as well. The pipeline finishes off with a comprehensive report. The report has 3 sections. Here you see the sequencing and mapping quality control section. I would like to highlight the histogram showing us the number of reads per sample in the various analysis pipeline steps. In the exploratory analysis section, he can explore the similarity between the samples, either by correlation or by principal component analysis. The differential gene expression section has plots such as a heat map for the differential expressed genes and a table consisting of the differential expressed genes per contrast, and also links to perform a pathway analysis, enrichment analysis. So we also have, in the end of the report, a method section depicting all the commands that were executed in the pipeline with links to intermediate and final results. I need to state also that all the outputs produced throughout the pipeline are organized in a structured folders according to the pipeline step. The epigenome pipeline basically consists of two workflows, one for ChIP-seq and one for ATAC-seq. The common thing between the two pipelines is that you start off with a FASTQ file, trim the reads, map to the genome, identify peaks, annotate peaks, and finishes off with a report. In addition, there are some QC tools run and genomic enrichment analysis tools on the peaks. So this is a section of the ChIP-seq report where we can see enrichment of genomic regions of the peaks and a table depicting the peaks per pair of IP sample and versus control as defined by the user in the interface. So the UTAP architecture is based on a singularity container that basically encapsulates all the necessary software. When the user submits a pipeline via the interface, the appropriate snake-make is executed. Within the workflow, parallel jobs per sample will be submitted to the cluster. This setup also includes a managing and monitoring system. UTAP can be deployed. It's free and ready for you to use on a local server or in a cluster or on Google Cloud. All the details of how to install it are found in read the docs. So to summarize, UTAP2 is a transcriptome and epigenome, contains a transcriptome and epigenome pipelines. It's web-based interactive interface. It contains comprehensive reports and structured outputs. It's open source and free, and it can be deployed on a cluster server or Google Cloud. It contains also a management and monitoring system. So I would like to acknowledge the people that contributed to the development of UTAP2, people within my bioinformatics unit, especially Jordana Lindner, which is the programmer. Please try out our demo site, which is open, and you can feel the interactive interface and the see reports for the various pipelines. And we're happy to answer any question and to assist you. And thanks for listening.
Questions?
Hi. So I see that you can deploy this on the cloud.
Right.
Does it also, can you actually use files that are located on the cloud to
Yeah.
load up?
Yeah.
Okay.
You need to load your sequences to the cloud. Yeah.
Oh, so that's essential.
Yeah, of course.
Got it. Thanks.
Question. Could you give us an idea of number of users that you have using the system and how
Okay.
is the adoption?
The adoption is very, very simple. Like I showed you, the interface is very simple to execute. Basically, we also use it in order to teach the students. So it's part of our course. They can execute simply all these pipelines and explore the results. And so we have hundreds of users over the years, only within Weizmann Institute. It's very, very popular because it is so easy to use.
Nice talk, very useful too. So I have a question. So you guys have thinking about put some single cell process side?
Yeah. Yeah, so initially we wanted to integrate at least Cell Ranger as a starting point. I would say that the problem with integrating Cell Ranger is that it's updated so often that it's a race. And also people need to be very understanding about if they're only running JEX or running JEX with attack or otherwise, because its command is different. The genomes get updated very often, and the software gets updated very often. So at this point, I thought it was not stable enough to integrate it inside.